So what are some applications of DNA technology? Applications, DNA technology. All right, well let's first look at medicine. So what are some applications of DNA technology in medicine? Well, one of the two big things where recombinant DNA technology was first used was to create insulin and human growth hormone. Human growth hormone. So before the advent of recombinant DNA technology, insulin and growth hormone were really hard to manufacture. You basically had to isolate it from another human and purify it and then give it to patients. But with recombinant DNA technology, you can basically just grow these proteins in E. coli. You can grow them and culture them in E. coli bacteria. So this really has changed the way that medicine is practiced, and it's really helped a whole bunch of people. So vaccines is another application of DNA technology. A while ago, vaccines were made by first denaturing the disease, and then after the disease has been weakened, they would inject it into a human, and they would hope that their immune system would be able to put up a fight against the weakened virus. And that way, in the future, if they were infected with that virus, they would at least have some kind of immune response towards the virus. The problem with this was that the patient would sometimes get the disease because you're injecting a weak virus, but sometimes it wasn't weak enough. So with DNA technology, they can actually recreate the outer shell of the virus and inject that. So it's a lot more cost effective, and it doesn't have the risk of actually causing the disease in the host. So this is much safer, and it is cheaper, and it's it produces a better immune response. And so some vaccines that we actually use recombinant DNA technology to create include the Hep B virus and Hep B virus and the herpes virus and malaria. So these are some applications of DNA technology in medicine. Another cool application of DNA technology is in solving crimes. So solving crimes. So in forensics. So there are parts of the genome known as non-coding regions of the genome. And these regions can actually help forensic scientists identify specific individuals. So they can look at things like short tandem repeats, STRs, and these are basically short sequences of DNA, like 2 to 6 base pairs long, and they're normally found in really high amounts. They're just these short repeats that are found in really high amounts and to varying degrees between different individuals. So if they actually sequence these short tandem repeats, they could identify specific individuals given a DNA sample. They can also look at mitochondrial DNA. So mitochondrial DNA is inherited from your mother, and it's found in really high amounts within an individual cell. So even if there's very little sample available, forensic scientists can analyze mitochondrial DNA in order to identify a potential suspect. Another technology that is used in forensic science is Y chromosome typing. So that's basically YSTR. And this is looking at short tandem repeats found on the Y chromosome. And so DNA technology has helped scientists pick out individuals that have committed various crimes based on DNA samples that they were able to find. So agriculture is another field that has greatly benefited from recombinant DNA technology. So, for example, scientists can now create plants crops that are resistant to insects and that are resistant to herbicides, and can also delay ripening of the crop so that you can transport the crop from the farm to the store. So by doing this, you're basically able to create more crops to feed a growing population of individuals. And it also helps with the economy because then you've got farmers that are growing all their crops, and if there was some sort of bacteria or virus that destroyed their entire crop, then that farmer would not get paid for that season. So by transgenically modifying the crop so that it's resistant to specific things, then they're able to grow their crop, sell it, and feed individuals.